Okay, welcome back from that really long break. Now it's the second half of the semester. So remember, if you picked up your exams, or if you haven't yet, please pick them up, or they'll be in the lab later. I threw out questions 7, 11, 27, and 30. So make certain that my arithmetic is correct. You should see some kind of a mark if you got any of those wrong, and you should have the points added back. If I missed that somehow, please let me know. Happy to raise anyone's grades. If you accidentally got too many points, don't even bother to tell me. Just consider that manna from heaven. My math's not that good. Okay, so we left off last week talking about, what were we talking about last week? Oh yeah, we were talking about gene regulation, and we left off talking about riboswitches. So now we want to continue with this theme of gene regulation, but now we want to look at gene regulation, as I mentioned last week, on steroids. So now I want to look at what are referred to as global regulatory systems. And in reality, you've already looked at one of them. So this is a table from a textbook of some of the best known global regulatory systems. So these are widespread. These are large numbers of genes that are regulated coordinately. We've already talked about the SOS response in response to damaged DNA, or actually also in response sometimes to antibiotics or nutritional stress, but certainly in response to damaged DNA. This global regulatory system is mediated by the repressor LEX-A, and more than 20 genes or operons throughout the chromosome are regulated. So in essence, I could really stop lecturing at this point. You already know how global regulatory systems work. But instead, I want to use what's probably really the king, or queen maybe, of all of the catabolite repression systems. Of all of the global regulatory systems called catabolite repression. If you look at this table, it's obvious to see that catabolite repression, which we're about to talk about, has the widest degree of control of gene expression in the bacterial chromosome. More than 300 genes in E. coli, genes and or operons, are regulated in this global regulatory system. We're going to talk about one called catabolite repression. It's probably also known better, and this term will actually make a little more sense to you if we refer to it by its alternative name called the glucose effect. In essence, we've alluded to this already. I've told you that when bacteria have the choice in the environment to use glucose or any other carbon source, they will always use glucose. There is no simpler, more efficient energy source for bacteria, like E. coli, to use than glucose. And you know why? Because if the bacterium utilized any other carbohydrate, for instance, it would have to convert that somehow to fit into the glycolytic pathway. Glucose, no need to do that. It goes in just fine, thank you. So this is now what we want, rather than have you just rely on the fact that you know that bacteria like E. coli will use glucose first, and you know why. Now I want to look at the mechanistic reasons. How is it that bacteria have evolved to be able to make sure that they use glucose to the exclusion of every other delicious carbon source in their environment until glucose is completely gone from the environment? That's what the glucose effect is, also known as catabolite repression. Now, you didn't think we were done with the lac operon yet, I hope. So the lac operon is what we're going to use as an example to show how catabolite repression works. And as we go through these remaining painful steps of the lac operon, remember that when we're talking about the lac operon, this repression that we're talking about, this catabolite repression, is not simply affecting the lac operon. It's affecting all of the other catabolite operons. That is, operons of genes that would break down sugars like mannitol or mannose or galactose. All of those genes that would be required to break those down are going to be repressed the same way that lactose is. But lactose is the best studied of these systems, so we'll work with lactose. So remember, as we go through this glucose effect or catabolite repression, whichever term you prefer to use, this repression is being mediated by a secondary messenger that you're all familiar with from some level of biology class called cyclic AMP. This is something that's used in your cells and my cells as a secondary messenger, but bacteria like E. coli, not all bacteria, but a large number of them, including E. coli, use cyclic AMP to control gene expression. And in this particular case of this glucose effect, this repression of the lac operon, and therefore all of the other catabolite operons, like mannitol, for instance, are controlled by cyclic AMP levels. So if you look first at this graph that's shown here, this pretty multicolored graph, what you're looking at is over time in hours, shown on the x-axis, are either on the y-axis to the left, the number of bacteria changing over time, and to see that, we're looking at the green line. If you look at the number of bacteria that are growing starting at time 0, where the starting conditions have equal amounts of glucose and lactose, you're going to do this experiment in the laboratory, I hope, in a week or two. If this bacterium starts to grow in the presence of both glucose and lactose, the bacterium, after it goes through a lag phase, goes into logarithmic growth, and you see this maximal ramp, this maximal rate of growth of E. coli, the logarithmic growth phase. And at the same time, if you look at the levels of glucose and lactose in the cells, you'll notice that glucose drops precipitously, whereas lactose is unchanging during this initial period of time. This makes perfect sense. You all know that this is going to happen. E. coli is going to use glucose before it uses lactose. So what you see is that as glucose is depleted until it gets down to unmeasurable or below limits of detection, what happens now is that the E. coli goes through another lag phase. This type of a growth curve is what's known as dioxic growth phase, shown here at the top. So the bacterium in this particular case is going to go through a second lag phase. Now, at the end of that lag phase, the bacterium begins to grow again in exponential phase. Notice, however, the slope of this line. It's not accidentally a lesser slope than the first logarithmic phase. This is a real phenomenon. But what's happened during this second lag phase is the bacterium has run out of glucose in an environment that's filled with lactose, yet it doesn't yet have the machinery to utilize that lactose. It hasn't induced the lac operon yet. So that's what's happening in this second phase, is that beta-galactosidase and the other two genes that are part of the lac operon are being induced. So enzyme induction is occurring here. And then once beta-galactosidase is created, that lactose can be imported by the lac permease and broken down into glucose and galactose, and the bacterium continues now to grow. But notice that it can't grow quite as rapidly, because now it has to commit some of its resources into synthesizing the enzymes or all of the proteins of the beta-gal operon, and then taking the galactose that's generated from breaking down lactose and turning that into a form that fits into the glycolytic pathway. So some energy is being siphoned off now while it's using lactose. So while it can still grow just fine, it just can't grow just as fine as on glucose. So that's why there is a decreased slope to this. The bacterium will not grow as rapidly on lactose, simply because it has to invest more of its energy into doing that. So, as I mentioned at the beginning of this slide, remember that cyclic AMP, this molecule here, generated by an enzyme, a very important enzyme called adenyl cyclase, very similar to the enzyme in a eukaryotic system, but now present in the bacterium. Adenyl cyclase will take ATP and turn it into this cyclic AMP. And it's the levels of cyclic AMP that help determine the expression level of beta-galactosidase and the rest of the lac operon. But in reality, what's really going on here goes back to a principle that you already learned for the first exam, the phosphotransferase system. You thought that you could forget about it, but it's come back to haunt you. You still need to understand this phosphotransferase or glucose transport, because it is actually the process of transporting glucose from the environment into the cytoplasm of the bacterium that dictates how much cyclic AMP is present. So when glucose levels when glucose is being transported into the cell, by this phosphotransferase system, cyclic AMP drops. So when glucose levels in the cell are high, cyclic AMP levels are low, because adenyl cyclase, as you're going to see, is going to be inhibited by the phosphotransferase system. The process of importing that sugar turns off this adenyl cyclase, so cyclic AMP levels drop. And to make it even more exquisitely sensitive, the bacterium has the ability to take whatever cyclic AMP is produced and just kick it out of the cell. So that will be actively exported from the cell. So when glucose is being transported, in effect, cyclic AMP levels drop to near zero inside of that cell. High glucose transport, low cyclic AMP. Now, here's the lac operon and its control features. So this cyclic AMP molecule, a secondary messenger, is another example of an allosteric modifier. In previous lectures, we talked about how arginine could modify a protein, allolactose could modify a protein to affect its ability to bind DNA, negatively or positively. So here, cyclic AMP is a modulator of a protein called catabolite activator protein, called CAP. Back when I was in graduate school, they mistakenly called this the catabolite repression activator protein. But they soon realized that students were always going to call this protein CRAP for its acronym. So now textbooks almost invariably refer to this as the CAP protein, or catabolite activator protein. So here's the lac operon shown here on the top. And here are the genes for the structural enzymes, the beta-galactosidase or LAC-Z. The LAC-Y is the permease, and the LAC-A is the transacetylase. We talked about how this repressor protein, encoded by this gene shown here in orange, LAC-I, the protein produced from this gene actually binds to the operator. Remember, LAC-I protein is a repressor. It binds to the operator to try and stop transcription. And so when lactose is absent, or more specifically when allolactose is absent, this operator site is going to be, when that's absent, is going to be bound to the repressor to try and stop transcription. But if that bacterium should encounter some allolactose, and so the repressor comes off, that might, you think, allow RNA polymerase to bind to this LAC promoter and begin to transcribe the lac operon. And you're correct, but only to a small degree. So there are two conditions that are required for maximal expression of the lac operon. One is the presence of lactose, and then converted into allolactose to make that lac repressor drop off. But that's only one condition that's necessary for the lac operon to be maximally expressed. The other is an activator protein. This is the catabolite activator protein. So now what happens when E. coli has run out of glucose? There's no more glucose in the environment, so its phosphotransferase system is no longer importing that sugar. When that happens, cyclic AMP levels, which had been near zero, will now rise, because the inhibition of adenyl cyclase is removed. The phosphotransferase system has stopped importing glucose. So adenyl cyclase activity increases, and so does cyclic AMP. So if you think about the catabolite activator protein as a pool, it can exist in two forms. It can exist in a form where it's not bound to cyclic AMP, or one where it is bound to cyclic AMP. And as glucose levels have dropped, this pool of catabolite activating protein tends to shift to the right in this figure. So cyclic AMP binds, allosterically modifies that protein, so that now it has DNA binding activity. This is a classic activator protein. So this is going to bind to this CAP site. Notice the position of the CAP site. The CAP site is where the catabolite activating protein binds if it's bound to cyclic AMP. That's upstream of the LAC promoter. this is exactly where an activator in a bacterial transcription system should bind. It's going to bind here to help RNA polymerase bind to this lac promoter. And now we've already established the fact that now lactose is present, so there's not going to be a lac-I protein bound to this operator. So now you have both conditions satisfied. You have the absence of the lac repressor and the presence of the catabolite activator protein bound to cyclic AMP. So now you can have maximal expression of the beta-galactosidase genes, the entire lac operon. Again, let's look at two different situations here to try and illustrate these side by side. So on the left is a situation where the bacterium is in the absence of glucose, but the presence of lactose. And on the right is the presence of glucose plus the presence of lactose. So this would be early on in that growth curve that we were looking at. So under early conditions where glucose is present, glucose is being transported in, adenyl cyclase is being inhibited, and so there's very little adenyl cyclase. So the CAP protein, the catabolite activator protein, largely exists in the apo form, so the absence of binding to the catabolite, to cyclic AMP. And so it doesn't bind to an activator site. But in this particular case, lactose is present, so it can be converted at some small fraction into allolactose to bind to this, this tetrameric protein shown here by these 4 yellow balls, now bound to allolactose. That is the inactive form, so it can't bind to the operator. So you'll see some transcription going on, that's what's represented by this red line, and some amount of protein is present. So the bacterium, and it's important that this transcription never be completely stopped, right? Because if there was no transcription of the lac operon at all, then the whole system shuts down. Think about this. If there's no transcription, because this is a great exam question, if the whole system shuts down, you're no longer producing the lac permease, right? So if the bacterium does find itself in an environment where there's lactose, it can't import it. If it can't import lactose, the beta-galactosidase enzyme can't convert some of it into allolactose to now induce the transcription by getting rid of the repressor. So the situation is never such that the lac operon is completely shut down. Even when glucose is present, there's going to be, and there needs to be, some minimal level of the beta-galactosidase enzyme and the lac permease. Otherwise, the system is just never going to work. But now, in that growth curve that I showed you a couple of slides ago, you saw that once glucose was used up, but lactose was still there, what happens next is there's that pause, that second pause in the dioxic growth curve. That's representing what's happening here on the left-hand side of this schematic. The absence of glucose, so glucose has been utilized, but the bacterium has lactose present. Now, it was already producing a small amount of lac permease, so now the bacterium has the ability to import, using its proton motive force, import some of the lactose. It has some beta-galactosidase enzyme, so it has the ability to produce more of the allolactose, as well as to break down lactose. But now you see here, you have adenyl cyclase no longer being inhibited. So now you get that second requisite condition for expression of the lac operon. That is, adenyl cyclase is now permitted to be active. Cyclic AMP levels rise. So always remember, high glucose, low cyclic AMP. Low glucose, high cyclic AMP. So without glucose present, this high cyclic AMP now shifts the pool of catabolite activating protein more towards the bound form or the active form. You could use those two terms interchangeably. The bound form is now an activator. There is no repressor bound to the operator. So now the lac operon is going to be transcribed like a freight train. It's going to make lots of transcripts. It's going to make lots of proteins. But this is what was occurring in that second lag phase, was the shift from only a little bit of proteins from the lac operon to a lot. So this will now allow the bacterium to take off using lactose as this other carbon source. Now, mechanistically, how does the phosphotransferase system mediate this catabolite repression? And again, as I'm talking about this and using the lac operon, keep in mind that this is happening simultaneously for every other sugar that the bacterium could utilize. Glucose, remember, so just to quickly review, glucose on the outside of the cell, shown here in the top, is transported across and simultaneously phosphorylated into glucose 6-phosphate by this enzyme 2CB specific for glucose. So here is the phosphotransferase system again. The phosphoryl group is coming from phosphoenol pyruvate, going into enzyme 1, which phosphorylates this histidine protein. That's the non-specific portion of the phosphotransferase system. That's the system that functions to transport any sugar into the bacterium. Now that phosphate group, if glucose is present, gets transferred to the first of these glucose-specific enzymes, 2A glucose. That phosphate group doesn't stay there very long. If glucose is out here and being transported, glucose is actually acting as a phosphorus siphon. It's sucking up the phosphate groups as fast as it can. So as fast as these phosphates are coming in from phosphoenol pyruvate through enzyme 1, HPR, 2A glucose, it's not staying on 2A glucose for very long because it's being fed onto 2CB glucose, and then that phosphate is going on to glucose. So the predominant form of this enzyme 2A glucose, and this is the critical enzyme, so it's important that you remember this. This is the critical enzyme for catabolite repression. The phosphorylated form of enzyme 2A glucose is at very low levels when glucose is being transported. Makes sense. Glucose transport requires the phosphate flux to go through here very fast. So enzyme 2A phosphate, I'm getting a little bit ahead of myself. So this form in the unphosphorylated form does something really important in terms of preventing the bacterium from utilizing lactose. The non-phosphorylated form of E2A glucose in the non-phosphorylated form accomplishes something known as inducer exclusion. That term is very descriptive. It's actually excluding lactose from the cell. This enzyme in the non-phosphorylated form, think of it as a cork or a plug. It can literally stick itself inside of the pore of LAC-Y, so that even if lactose is present on the outside of that cell, it can't get in. There is an enzyme stuck on the other side, this enzyme 2A glucose, in the non-phosphorylated form, which is stopping lactose from coming in. This makes sense. E. coli is bringing in glucose. It wants to use glucose. If it lets lactose come in to any significant degree, that's apt to start transducing to a higher level the lac operon. So a way to control that is to stop the lactose from even getting in. Inducer exclusion. Now, when glucose gets scrubbed from the environment, so going forward in time, the E. coli has now sucked up all of the glucose from the environment. What happens now is that you have a shift in this enzyme 2A glucose pool. Whereas when glucose was being transported in, the phosphorylated form didn't last very long. But now, what happens is that you get a backup in the system. Enzyme 2CB glucose here in the membrane is phosphorylated because it's waiting for a glucose. But with no glucose coming through, there's no place for that phosphate to go. So any phosphate on enzyme 2A glucose is also stuck there. So the pool shifts to this enzyme 2A glucose in the phosphorylated form. That form, I got my bullets a little bit out of place. I'm going to back up a slide here to get to this point. The phosphorylated form of that enzyme is actually what activates adenyl cyclase. So adenyl cyclase, when glucose is coming in, doesn't have an activator, and so its expression level of cyclic AMP is relatively low. Now, when glucose is gone, and enzyme 2A glucose in the phosphorylated form accumulates, that's actually going to cause cyclic AMP levels to rise, because this form actually activates adenyl cyclase. And I think you can also look at it as, and I've seen it written this way, that the non-phosphorylated form of glucose actually inhibits adenyl cyclase. I think both of those statements are actually true. But in either case, you can see why in the presence or in the absence of glucose, you want cyclic AMP levels to rise, because cyclic AMP levels are going to allow the transcription of the lac operon if lac is present. The other thing, that really cool thing that happens when glucose is gone and enzyme 2A glucose shifts to the phosphorylated form, that phosphorylated form can't fit into lac-Y. Inducer exclusion stops at that point. That cork can no longer fit into that pore, and so now lactose can be transported in if lactose is present. This is going to be happening on all of the other permeases for sugars, for mannitol or for whatever sugar or carbohydrates. So this will allow inducers to come into the cell if they're present. So lactose is now free to induce the lac operon. It gets rid of the repressor. You also have high adenyl cyclase levels going on. So that's the two conditions that you need for maximal expression of the lac operon. So this is just another cartoon that I think helps to show this process side by side. And maybe one of these explanations resonates a little bit better with you, because it's important that you understand it. On the bottom here is a situation where you have catabolite repression. So glucose is being brought in and turned into glucose 6-phosphate. In this particular case, adenyl cyclase levels or cyclic AMP levels are going to be dropped, going to be near zero, because adenyl cyclase is being repressed in its activity. And so what's going to happen is the non-phosphorylated form of this enzyme 2A glucose can actually do inducer exclusion. So it's shown here as interacting with the permease. This would be the lac-Y gene product that stops lactose from coming in. Now, the situation, so now let's say the bacterium has removed all of the glucose from the environment, scrubbed it completely. But now it's in an environment that it has lactose or maltose. We're just dealing with lactose here. Now, adenyl cyclase is now active, because instead of having the non-phosphorylated form, which perhaps inhibits the adenyl cyclase, it also has the phosphorylated form of that same enzyme, which is an activator of the enzyme. So it actually binds to the enzyme to help it take ATP and turn it into cyclic AMP. Cyclic AMP levels rise. It binds to, this is an older slide, this is what they used to call it, the catabolite repressor protein, and sometimes the catabolite activator protein. But it's that CAP protein. So now the pool shifts, and you have predominantly bound form of the catabolite activator protein, which binds as an activator to all manner of operons encoding catabolite pathways, such as the lac operon, such as the maltose operon as well. So this is mechanistically how catabolite repression and the phosphotransferase system are linked. For time purposes, we're going to skip this one. I'm sure you'll all be really interested in the comparison between how gram-positives and gram-negatives do it. But there won't be anything on an exam about this. Okay, the last topic that I want to cover under gene expression is one of my favorites, maybe is my favorite, and that's two-component signal transduction systems. And again, this is a system that you already know a fair amount about. We've touched on this before when we talked about chemotaxis, when we talked about quorum sensing in gram-positive bacteria. So you have seen some manner of this two-component system before. But this system that we're going to describe and spend some time talking about now is the most widespread means by which bacteria sense changes in the environment and transduce that information into the cytoplasm so they can adapt to those changes. The other really cool thing about these two-component systems is, like peptidoglycan, this is not found virtually anywhere else except in bacteria. So there are a few fungi There are a couple of plants that have something that resembles two-component systems, but your cells and my cells and all of your pets' cells, anybody, all of these multicellular organisms do not have two-component systems. This is part of the reason why this is really interesting, because no one has yet managed to bring a drug to market, so far as I know, that targets this two-component system. But if one of you is clever enough to come up with a way to interfere with two-component signal transduction systems that we're about to describe, there probably would not be ill effects on you or I, because we don't utilize this kind of a signal transduction pathway. It's exclusive largely to bacteria. That's part of the reason why it's important to understand this system. Although, for the life of me, I still can't figure out why no one has designed a drug that targets these things. So there must be something that I'm missing in this puzzle. But this is the system that I'm about to describe is very similar to the system when you go back in your notes to study quorum sensing. When you look at how quorum sensing occurs in gram-positive cells, not gram-negative, but gram-positive cells, you'll see some similarities. It's a two-component system, so let's start with the first component, which is known as the sensory component, or the sensory arm. It's a membrane protein that spans the cytoplasmic membrane of the bacterium. This could be gram-positive or gram-negative. It has a domain which exists in the periplasm, or the extracellular environment, which is responsible typically for binding some type of environmental ligand. This could be a salt molecule, this could be a sugar molecule, this could be an amino acid, this could be a toxin. It could be nothing at all. Some of these sensory kinases, this protein here, actually sense heat or cold. So obviously that's not an environmental ligand. But they have an input domain in which they interact with the environment. A conformation change occurs when this protein interacts with either a ligand or some kind of an appropriate signal. Maybe it's temperature. The conformation in that protein alteration is now occurs on the cytoplasmic side, so that now the protein becomes an active enzyme. This protein is a histidine kinase, and we've talked about this. Key A in the chemotaxis system was your first exposure to a histidine kinase. This is slightly different in terms of its location. Remember that the key A protein was entirely cytoplasmic. It interfaced with the exterior environment only by means of that methyl-accepting chemotaxis protein. That's a very unusual arrangement for histidine kinase. Every other histidine kinase that's part of signal transduction pathways has an arrangement almost identical to this, where it itself spans the membrane. So it senses an environmental change, and that environmental change will activate the kinase activity on the cytoplasmic side, which will put a phosphate group onto a conserved histidine amino acid. It's always a histidine amino acid that becomes phosphorylated. And histidine phosphorylation, again, is something that's relatively rare, if not very rare. Yeah, I would go so far as to say very rare to almost non-existent in eukaryotic cells. Histidine phosphorylation is almost exclusively a bacterial and archaeal system. So this phosphate group can now be transferred to a response regulator. And again, you've talked about, or you understand response regulators. You've talked about how key A could phosphorylate key Y, could phosphorylate key B. Those are response regulators. But remember, in that system, those response regulators interacted with another protein. They interacted either with the MCP protein or the basal body of the flagellum. Again, the chemotaxis system is a very unusual signal transduction system. Two-component systems in general work the way I'm outlining them here, where the phosphorylated response regulator now does not interact with another protein. Instead, it interacts with DNA. So typically what happens is that the response regulator is an inactive protein until it becomes phosphorylated, and then it usually becomes a dimer and binds to DNA, to specific DNA sequences throughout the chromosome. So what's being shown here is just one particular site where a phosphorylated response regulator now becomes active and it binds an operator which stops the transcription of those genes. But this would be happening at 10 or 15 or 100 different genes or operons throughout the chromosome simultaneously. And only some of them would be binding to an operator to stop the transcription. In some other cases, it would be binding to an activator to turn on the transcription. And if you think about it, this makes sense because this is all occurring in response to a change in the environmental concentration of some signal. So if the bacterium needs to adapt to that stress or adapt to that bonanza, maybe if this is a nutrient, it needs to now change its gene expression pattern in order to adapt to that change. So this is the way bacteria tend to do this. And virtually all bacteria have multiple two-component systems to do exactly this. So that phosphoryl group is transferred from this histidine always to a conserved aspartate amino acid on the response regulator, and then that protein can become activated only when it's phosphorylated. Because remember that a phosphate group is highly negatively charged. It has a very high energy bond here with this aspartate. So it's very good at changing the conformation of that protein. So it changes it to an active conformation. And again, just to draw some contrast and to refresh your mind, because again, you thought you had seen the last of chemotaxis. Sorry. Remember that the histidine kinase PA could phosphorylate PY, which was now an active response regulator, but it has nothing to do with transcription at that point. No gene transcription is altered in chemotaxis. Instead, the phosphorylated PY, now active, interacts with the flea protein here in the basal body to stop the counterclockwise rotation and turn it clockwise. So chemotaxis is a very different type of signal transduction system. And so as we talk about specific signal transduction systems, this is just a partial list of systems that are found in E. coli. E. coli probably has about 20 of these systems, some of which we have no idea what in the world they do. So we've already talked about the Ka, Ky, and B system in chemotaxis. So we're going to talk now in detail, just to illustrate the points of two-component signal transduction. We're going to talk about a system here on the top, where the histidine kinase is a protein called ONZ, and the response regulator is a protein called OMPR. These are proteins that mediate E. coli's ability to go from a high osmolarity concentration environment to a low osmolarity concentration environment. Now, let's just go through a generic primer on how this two-component systems work at a little bit more detail than that last slide. So here is the periplasm of an E. coli cell. These little lines are meant to represent porins, so you remember that the periplasm is kind of a microcosm of the environment, so small molecules can diffuse in. Here's the histidine kinase molecule. It doesn't act in isolation. It actually acts as a pair. So here is the signal input domain in the periplasm. But ONZ and virtually all histidine kinases work as a homodimer, so two identical proteins. And actually what's going to happen here in ONZ, so ON stands for envelope. So when this was first discovered, it was known to be some type of a signal protein that was found in the envelope of bacteria. OMP-R is the response regulator. This is the second component. OMP is an unfortunate name for this because 99 times out of 100 in bacteriology and microbiology, when you see the abbreviation OMP, it stands for outer membrane protein. In this particular case, this is an historic term, so it's been maintained. It actually stands for osmoregulated protein. So this OMP actually preceded the designation for outer membrane protein. So it's a little bit confusing. But this is the response regulator. So what happens when E. coli senses a change in solute concentration? This is an example of a histidine kinase that is not binding a specific ligand. It's not binding to one amino acid or one toxic molecule. It's somehow sensing the overall solute concentration. So it's possibly membrane turgor. So as the bacterium is at a high salt concentration versus a low salt concentration, the osmolarity pressure to burst that cell or shrink that cell tends to change. So some people believe that occurs. But this signal input domain is critical to sensing that change in solute concentration. So as solute concentrations increase, That information is transduced into the cytoplasm where you actually have not autophosphorylation, as it's usually referred to even in the textbooks. It's actually more appropriately referred to as transphosphorylation, because this is why it's important that there be two histidine kinase proteins, because in effect, one histidine kinase protein phosphorylates the histidine of its partner, whereas its own histidine becomes phosphorylated by its partner. So they phosphorylate one another. So that's kind of a minor point. But then that phosphate group is going to be transferred from that histidine to an aspartate. That's what D stands for on the response regulator. And now this is still a high energy bond. So there's a big change in the conformation of OMP-R. So now it has the capability of forming a dimer itself. It's normally in the inactive state, in the non-phosphorylated state, non-phosphorylated and a monomer. Now it becomes a dimer, and now it can actually express its DNA binding capabilities. HTH down here stands for helix turn helix. That's a very common DNA binding motif that a lot of proteins have. But you'll notice that, again, this dimer has bound into the major groove of protein, so it's binding most likely to an inverted repeat in here. But now, by being bound to this DNA, it can either stop transcription or promote transcription, depending on its relative juxtaposition to the promoter. So now that can activate the transcription of genes or repress the transcription of genes. But now what happens when the bacterium now goes from a high solute concentration environment to a low solute concentration? So I made the little lightning bolts disappear to indicate that now the environment has changed back. The bacterium needs to adapt to that. But unfortunately, it's got these covalent bonds between phosphate groups and this OMP-R, which would continue to repress genes or activate genes, which are no longer needed because the bacterium is now in a different environment. Many two-component systems, not all, but many two-component systems have a second enzymatic activity that doesn't get very much press. It's a phosphatase activity. So many times, the histidine kinase, in the absence of a signal, is not turning on its kinase activity so much as it's turning on the alternate activity of phosphatase. So now, as that bacterium went from a high solute concentration to a low solute concentration, it turned off its kinase activity, largely, and turned on a phosphatase activity, which is specific for that aspartate on OMP-R. And so now OMP-R falls off of that DNA, and now the bacterium goes back to what was its original expression pattern, now hopefully adaptive to that low solute concentration. Now, just to draw a little bit of contrast between eukaryotes and prokaryotes, this is for the biochem people out there, is that, we all, you and I, in our signal transduction systems use phosphorylation predominantly of serine, threonine, and tyrosine. Bacteria do this too, to a lesser extent, I think, than we do. But these phosphates are all phosphoesters. These are low energy bonds. So as you study signal transduction in cell biology, what you'll see is that the phosphorylation of serine might trigger another protein to become phosphorylated. But that's a separate phosphorylation event. That requires the cleavage of another ATP. This phosphate group does not move onto the next component in that pathway. That's very different biologically than what happens in bacteria. In bacteria, when the histidine becomes phosphorylated, this phosphoryl group is going to be transferred to the response regulator D, or aspartate. And it has to do with the difference in the nature of the bonds. So phosphoester bonds are very low energy. The phosphoimidazole bond in particular to this histidine molecule is a very high energy bond. Breaking that bond releases enough energy that it can actually help to mediate the transfer to this phosphate group, which is a very different bond as well. It's an acyl phosphate bond. Still a high energy bond. So very different biochemically than the signal transduction systems in eukaryotic cells. Now, let's dig in a little bit in the last 6 or 7 minutes to how it is that E. coli mediates the ability to move from an aqueous environment to the environment that it prefers in your colon or my colon. If you think about it, people don't think about this very often, but E. coli, like all of the bacteria in our intestinal tract, actually have to be able to survive in two different environments. Think about this. So the colon is where we think of E. coli living, but think of every time you flush the toilet, you're introducing billions of E. coli and billions of other bacteria into now, in essence, a freshwater environment. That doesn't kill the bacteria. From the bacteria's point of view, it's just thinking to itself, I can't wait to get back into a colon. I just have to survive in this aqueous environment for a while, but I want back into a colon. I don't care whose colon I get into. I just need to survive in this water. So what we're really talking about here is how it is that E. coli, and you can substitute virtually any other bacterium that comes out of your colon and into the water environment, survives and perhaps even reproduces in an environment until it can get back to its favorite place in the colon. It does this. The adaptation of E. coli between freshwater environment and the colon environment all hinges on the expression of two outer membrane proteins. This is why I talked about OMP-R as opposed to not being an outer membrane protein. Now I want to talk about OMP-F and OMP-C. With these proteins, OMP does stand for outer membrane protein. Outer membrane protein F and C are usually not expressed at the same time in E. coli. One is usually expressed to the exclusion of the other. The way that you can remember, so this is my way of remembering which protein does which. OMP-F, which has a larger pore, is the porin that is adaptive for freshwater environment. So this is the outer membrane protein that E. coli wants to express if, after you flush the toilet and it's now trying to survive in the water environment. Because in that environment, nutrients are at perhaps a premium, right? So in order to get those small molecules across into this periplasm where they can be subject to active transport, it's better to have a big target, a big hole, a big pore. So the pore size in OMP-F is 1.16 nanometers, as opposed to the outer membrane protein C. C, think of as standing for colon, because OMP-C is the protein that E. coli wants to express in a high solute environment, such as the human or the vertebrate colon. It has a lower ion flux rate because it has a smaller pore size, 1.08 nanometers. So it's a smaller target for small molecules to fit across that pore. That makes sense in the colon because some of the small molecules you don't want to come across because some of them are going to be toxic. Bile salts and things like that could kill the bacterium. So in an environment where there's lots of nutrients but lots of toxins, the bacterium wants to be a little bit careful about who it lets into its periplasm. So it's best for that bacterium to express OMP-C. So it's the ability to regulate which of those proteins are expressed that allow the bacterium to survive in these two different environments. And that all has to do with this two-component system on Z. So here's the histidine kinase in the cytoplasmic membrane of the bacterium. And here's OMP-R, the response regulator. Remember here, OMP is a DNA binding protein that stands for osmoregulated protein. When the bacterium, let's start with a situation where E. coli is in a water situation and it's dying to get into somebody's colon, just hoping. In that particular environment, the osmotic pressure is going to be low. There's not going to be a lot of solutes there. So that ON-Z protein is going to be relatively kinase quiet, not silent. It's important that you realize that the kinase activity, even under low solute concentrations, is never zero. It's always going to be phosphorylating at some random firing at some random small level. What that's going to do is that's going to make the phosphorylation of the response regulator quite rare. So if you think about this response regulator, this transcription factor, OMP-R, as a pool, as I often like to explain it, you can think of that pool as shifting between the two forms of phosphorylated versus non-phosphorylated. In the aqueous environment where pressure is low, osmolarity pressure is low, there's going to be relatively low levels of OMP phosphate and relatively low levels of OMP-R non-phosphorylated. Now, it is the phosphorylated form that's active. Here are the genes for those two crucial outer membrane proteins, OMP-F and OMP-C. If you look at the promoter region for OMP-F, This is the gene that needs to be expressed if this bacterium is going to survive in an aqueous environment. There are two sites where OMP-R can bind. One is high affinity and one is low affinity. So the key is down here. The checkerboard, the high affinity is, in this case, the lightly stippled area. That's an activator site. If it binds to that high affinity site, it's going to turn on OMP-F. And if you think about it, when OMP-R phosphate is at low level, where is it going to bind? It's going to bind to the high affinity site, and it's going to turn on these genes. I find myself accelerating and accelerating trying to get through this. So what I'm going to do is I'm going to take a breath. We're going to stop for now. I'm going to finish these slides on a separate screencast. So I'll put this screencast up, and then from home, I'll go through this whole system again a little bit more slowly, a little bit more carefully, and I'll post that one as well. So the exam material ends at the end of this slide set. So we haven't quite finished with this two-component system. Okay, thanks guys. I'll see you on Friday.